Yeah, so I'm recording, so you're on cast. You're on cast. Recording and recording. From here? Yeah, I'm recording. There's still people coming in. So staff deals with you. So oh, that's great. I will take down. Yeah. I mean, it's I didn't realize you were. So I just. But yeah, that's great. Get out of the state. It's hard for to get out there. Testing. Can you hear me up the back? So we'll get started, everybody. Thank you all for coming. We have a special guest who's come a long way to speak with us today from over the river. I actually live even closer than I work, so it was just over the Muddy River. Yeah, well, we invited you to save money on travel costs, but we also invited you because of your many achievements, which I'm going to read some of now. So if you don't know Manolis, the first thing to know about him, he's extremely gregarious and caring as a human being, which is something you really appreciate when you get to know him. But he's also extremely intelligent and used his ability to network to be able to bring together really big and successful teams over his whole career. And so just to give you the formalities, he trained at MIT. His undergrad, he has a degree in engineering and a PhD in computational genomics with Eric Lander and Bonnie Berger. He's now at the Broad Institute and is a faculty member over the river. He is an associate member of the Broad, a PI at the Computer Science and Artificial Intelligence Lab, and is head of the MIT Computational Biology Group. His work spans really a lot of things, as you'll see today, with a focus on human disease, actually, disease circuitry, genetics, genomics, a big focus on epigenomics. He was led the ENCODE project, which you may know from a number of years ago was the first major roadmap of the human epigenome. He also was involved in the GTEx project, the Genotype Tissue Expression Project, and a lot of other things. I won't chew up the time because I know he has a lot of things to present today. He's won many awards, including U.S. Presidential Early Career Award from the President Barack Obama, and the NIH Director's Transformative Research Award, and was recognized as one of the Technologies Review under 35 stars, among many things. And I'm amazed at his productivity. This is something that's rare for someone his age. And we just found out that he's, what, about 8 years younger than me. He's published already.
Still remain undisclosed. On what you're working.
It's published, this may be out of date, but close to 250 articles already, cited 138,000 times, quite impressive. And so today he's going to tell us about his latest work. I happen to notice and confirm with Manolis that in the last week or so, he's published a Nature paper, a Cell paper, and a Nature Communications paper, which really is quite the achievement. Today's seminar is up there, the title, which is AI, a hot topic, for genomic medicine, cellular circuitry, protein drug, function landscapes, and patient trajectories. Thanks, Manolis, for coming.
Thanks so much. It's really a huge pleasure to be here. I'm in this building very, very often. I live, as I mentioned, just across the river, and across the Muddy River, and I work across the other river at a small school. It's a technical school affiliated also with Harvard. When I was an undergrad, my friends used to say, "Mom, don't you understand? It's the sister school of Harvard. I can take any class I want there." And to which moms would respond saying, "You know what? If I were you, I would take all of my classes at Harvard." So anyway, I'm from this technical little school across the river. So I'm here to share my excitement for where the field is heading. I think that this is an unprecedented time, not just for all the horrible things that are happening in the politics, but for the extraordinary things that are happening in science today. And I think that we are close to really understanding a unification of biology, a unification of medicine, being able to understand biology, chemical structure, protein function, cellular space, physiological space, disease state at unprecedented levels of resolution, at a level that humans alone would never be able to do. That's why I put AI in my title, because I feel that the representation learning that we're able to carry out right now with some of the latest techniques that I'll talk about is transforming my own understanding and my own view of the natural, physical, biological world around us. And I think that it is giving us access to insights that we would never be able to reach otherwise. And I think combining that with the extraordinary technologies of genome sequencing, of high throughput perturbations, of profiling, of integration, and also with the extraordinary automations that we've seen and the plummeting costs of sequencing, I think is the golden age of biology that we're living through right now. So I'm truly just absolutely delighted to be here in such a time, bringing together our sister institutions basically between the medical, the technological, and the biological. So what I'll talk to you about is how we're using AI to understand the molecular circuitry underlying human disease and human biology. How do we go from genomics to understand the wiring, to infer targets for manipulating disease, and ultimately design drugs, and of course test them in animals and in patients. And in the area of healthcare, how do we apply that to understand patient heterogeneity, patient trajectories, and how do we characterize patient in multimodal ways across text and structured data? And also, how do we build foundational AI techniques for drug discovery, for understanding protein function, and for understanding patient states? So since my birthplace of Greece and the ancient Greek view of medicine, of having some kind of god or temple physician, the distinction was subtle back then. I think still is today. You would sort of look at the patient from the outside, And this all changed, of course, in the Renaissance with the advent of microscopy, of being able to actually look inside the body and look at individual neurons. These drawings are from more than 100 years ago. And in fact, even this picture is from 115 years ago, from the first diagnosis of Alzheimer's disease, where you could basically see that what we thought was some kind of, I don't know, exorcism-worthy malfunction of the brain could actually be traced to very clear cellular signatures of amyloid plaques and neurofibrillary tangles that were the initial diagnosis of Alzheimer's then, and is still the diagnosis of Alzheimer's today, 115 years later. And it's still the only thing that we actually are able to therapeutically target, just like get rid of the plaques. And unfortunately, we still don't know if that actually improves cognition or not. We're going to have to wait a few years for that. So it is incredible how far we've come since that time. And on one hand, we're here, of course, in a genetic seminar, and the genetics of human disease have given us more than 200,000 genetic loci across the genome that are causally implicated in disease from common variants alone, and thousands more from rare variants in protein coding regions. And we now have the ability to now start understanding the wires that connect these genetic variants that have causal effects to the actual phenotypes. So that's what I'm going to talk about, of course, for the first part of my talk, of how do we understand the wiring and use that to understand the molecular basis of many disorders across many loci. And in the second part of the talk, I'm going to talk about how AI is helping us understand the latent representations of biology. On the bottom here, on the bottom left, I'm showing you 4 cells. On the bottom right, I'm showing you 2.4 million cells. This is from one patient. This is from 430 donors across multiple regions of the brain. And every dot here represents 20,000 variables, the expression of every single gene in the genome. So how do we use AI to bring order to these massive data sets? Sorry, who was that? I don't think anybody's on Zoom.
Well,
There you go. Meeting's locked. Mess with me, you're out. So that's our goal today. So basically, how do we understand, number one, these paradigm shifts of going from hypothesis-driven research to data-driven research, where you used to formulate a beautiful hypothesis, gather data, do a lot of thinking before, and then do a very targeted study to get a very distinct and clear answer. And there's absolutely lots of beauty in that. But in the end, there's very little novelty that comes beyond your initial hypothesis. And of course, NIH always forces us to say, what's your hypothesis? Our hypothesis sometimes that if we gather this amazing data set, we're going to find awesome stuff. The data-driven research basically flips it around and says, listen, eventually we're going to need this type of data for every gene in the genome, in fact, for every cell in the human body, and in fact, for every person, in the world. So let's gather all the data, like shoot first and ask questions later. Building systematic data sets has been a lot of the focus of my career across many of these consortia, Roadmap Epigenomics, GTEx, ENCODE, et cetera. And these have proven to be extraordinarily useful, immensely helpful to the community and to many grad students and undergrads, postdocs, trainees across the world who do not have the resources to generate the massive data that you can generate, I guess here at Harvard before current situation. Share these resources massively across the world and build these comprehensive data sets. The second thing that genetics allows us is, of course, to go from correlation to causation. Instead of saying, people who drink more coffee have also better health, or countries that eat more chocolate per capita also have more Nobel Prizes, where epidemiology has always been about correlations. Is it because the coffee is really that great for you? It might be. Could also be that if you can afford, I don't know, $10 Starbucks coffee every day, you can also afford better healthcare, better nutrition, better lifestyle, etc. Causality-based analysis comes from genetics, and of course, genetic variants that are correlated with disease outcomes give us a one-directional arrow instead of a bidirectional arrow. So these unidirectional arrows from genetics is extraordinarily powerful for causality. And of course, even with polygenic risk scores, where you can actually calculate the risk of a person at birth or at conception and look at their correlation with manifestations that happen at 93 years of age, you have still causality. And then lastly, with perturbation experiments, it's much easier to intervene in a cell than it is, I don't know, in a social experiment with, let's, I don't know, get all the kids out of school for a year and see what happens. Little harder there. So that's sort of the beauty of causality there. And then the third major paradigm shift, which I'll focus the second part of my talk on, is going to be going from classical data analysis, where you have a new methodology for every problem. The human scientist does all of the thinking, and you're trying to minimize the number of parameters and build very targeted models, to a system where you build foundation models with billions of parameters, where you have multimodal representations across protein structure and chemical structure and protein function and text and so many different other facets of biology that are coming together in these unified representations. And I will dive really into representation learning as we start the second part, these hierarchical multimodal representations that truly allow us to understand concepts and give us dramatically new insights. So this is what we're using in my lab to basically dissect the mechanisms of disease-associated regions. We're basically starting with genetics. In this particular example, I'm showing you ApoE, the strongest genetic association with Alzheimer's. And we're looking at common and rare variants, of course, and we're starting with these regions of association. But the problem is that the vast majority of these regions are non-coding. 93% of genetic variants associated with disease are non-coding. So how do you make sense of them? Well, you need to understand the circuitry. You need to understand how these non-coding variants link to their target genes, what cell types they're active in, what are the upstream regulators, under what conditions do they turn on and off, and do they exert their action? When do they actually have an effect? And for all of that, we have basically needed to build these massive projects, such as the Roadmap Epigenomics project, the ENCODE project, the EpiMap project, and so on and so forth, to basically profile both RNA and the epigenome in health and disease. So the epigenome is made of three different signatures. Number one, the accessibility of DNA. That tells you if a regulator will be able to squeeze in and bind there. Number two, the modifications of the DNA, particularly CpG methylation, that basically sometimes changes the binding of regulators that only recognize methyl C instead of C, and vice versa. And then by far the richest and most complex, histone modifications, being able to see the post-translational modifications of the histone proteins that DNA is wrapped around, and understand at the level of about 150 bases to 200 bases at a time, the decorations of the genome that distinguish enhancers, promoters, transcribed regions, repressed regions, and so on and so forth. So first step is genetics. Second step is figuring out the circuitry. And the third step is massive data integration to predict driver genes, regions, and cell types. Of course, the fourth step is validating these predictions in human cells and in mouse models. Of course, disseminate the results and start the cycle all over again. So what do I mean by circuitry? I mean that we are going from a region of association to the specific genetic variants. And those variants are not anywhere. They're sitting in very specific enhancers. These are the control regions that can act at a distance and that target very specific genes. So if the genetic variant here is the causal one, then this is probably the causal gene. If that's the causal one, then those might be the causal genes. Again, it doesn't need to be one target. Sometimes it's multiple. And then understanding the cell types and tissues where these enhancers are active. Understanding the regulators that are binding and controlling these enhancers, and being able to also recognize the specific words, the regulatory motifs that are sometimes disrupted by these genetic variants, either directly or indirectly. So we now have methods that are, again, unprecedented in being able to read the genome and being able to predict the impact of genetic variants, even if we've never seen them before. And that's where all of the deep learning and so on and so forth comes about. So that's what we need to do systematically to understand disease loci. And my favorite example is a paper that we wrote almost a decade ago with Melina Clausen's in collaboration, both here and in Europe, that basically looked at the strongest genetic association with obesity that sits in the FTO locus. So that association was discovered 12 years earlier. and had remained a mystery for more than a decade. And the reason is that it sits in the middle of this FTO gene. FTO was ironically named FATO because it was a very long gene. It has a very long intron. And of course, as soon as we discovered it was the strongest genetic association with obesity, it was quickly renamed Fat and Obesity Associated, or FTO. But it comes from FTM, FTN, FTO, FTP, which were the fused toes screen in mouse. It did not have an obesity phenotype. It was just fused toes. So it has some kind of developmental function, but nothing with obesity. And in fact, various perturbations of the FTO protein led absolutely no results with obesity. So the question is, what's going on? Could it be another gene nearby? And what we found is that, in fact, the FTO region, the reason why this gene is so enormous is because it has a massive regulatory element within it that controls genes that are more than 1.2 million nucleotides away and 600,000 nucleotides away. And those genes turn out to be master controllers of a process that we all know and love, known as thermogenesis. Thermogenesis is your favorite friend nowadays. It's basically how your excess calories in your diet can be burned by depolarizing the mitochondrial membrane, causing a gradient, a loss in the proton gradient, and eventually loss of energy as heat. That's how you lose energy in nature. Conversely, lipogenesis used to be your best friend back when we didn't have so much food and we walked a little bit more. So when you have excess calories in your scarce diet at the time, you're able to store them as these energy-rich molecules known as lipids stored inside your white fat cells, which are white because they're full of lipids, compared to your brown or beige cells, which are brown because of all of the iron in your mitochondria. So this process, it turns out, of the developmental decision between whether you become a fat-burning brown adipocyte or a fat-storing white adipocyte was in fact at the root of this. And we would never know if we didn't have the ability to infer the epigenome across all of these different cell types to show that in fact this was this giant region where there's a 50,000 enhancer, 50,000 long super enhancer, 50,000 long region of association and a 12,000 long enhancer that sits there and that is active specifically in the pre-adipocytes that make the decision in this differentiation. It is only active in the first 3 days of differentiation. After that, the genetic variant has absolutely no effect. So we would have never seen it if we only studied mature adipose tissue. It's by studying pre-adipocytes that we're able to see it because that was actually in our data set when we looked in an unbiased way at these 117 tissues in the Roadmap Epigenomics project. We then used the motifs to figure out that the upstream regulator was binding this AT-rich motif, super creatively named this AT-rich interacting domain factor 5B. And it is a repressor. It binds that motif in the middle of the super enhancer. It represses that super enhancer, which then shuts off IRX3. But when the motif is mutated from a T to a C for the carriers of the risk allele, and you're all going to look down now because I'm going to tell you that I'm actually a homozygous risk carrier of the strongest genetic association with obesity. So my entire family has been obese. My father, my mother, my brother and sister, they're both on drugs, and I'm just exercising and dieting. So I'm homozygous risk, and basically my cells are stuck in the store position. Whatever extra calorie I eat, I store. So that basically means that I have a propensity to obesity. And of course, everything is genetics and environment. If you understand your genetics, you control your environment. And, so I'm not obese. So when my cells basically, because they don't have a T at that location, when they have a C and I'm homozygous, ARID5B can no longer bind. So IRX3 and RXC get de-repressed, and then they shut off thermogenesis. So every calorie is stored. So why am I showing you all this? Of course, as an engineer, it's amazing to decode genetic circuits. It's like so much fun. I'm sorry to say, yes, we care about the patients, but the circuits are just so much fun. But of course, having the circuits means we can manipulate them. And that's what we want to do systematically. We can basically go and perturb the right places. There's this very, very famous story of this MIT professor who was hired to fix a factory. The factory was costing them, because it was broken, costing them $1 million a day. And then he shows up, studies, and then puts an X. and they go behind that X and they find, the problem and they fix it. And then he sends them the bill of $10,000 for the consulting. And they say, could you please itemize it? So he says, chalk, $1, knowing where to put the X, $9,999. So knowing where to put the X, that's the hard part. So knowing where to intervene. So that's what we can do now. We can basically say out of 3.2 billion letters in the human genome, that's the X. That's the place. And when we go and use CRISPR-Cas9 genome editing to take primary adipocytes from carriers like myself and switch them from the C to the T, what we see is that thermogenesis, measured here as oxygen consumption rate, uncoupled as percent of basal, it's completely restored. So the cellular phenotype is restored by a factor of 7 by a single nucleotide alteration. So this is remarkable. It basically tells us that it's not just the enhancer, it's not just the expression of the gene, but also the cellular phenotype that can be altered with a single letter. And at the gene level, we can go into mice and put IRX3 dominant negative constructs under the control of an adipose promoter. What we see is that the mice that start out leaner are unable to gain weight. You put them in a high-fat diet here, and then the normal mice gain weight. These mice are unable to gain weight. So when they're sleeping, when they're walking around, they're just burning calories. They can eat all they want. They can exercise as little as they want. They're just burning their calories in their sleep. So who would like some of that? Good. You know who you are. And we have some in the back, by the way. I'll show you afterwards. I mean, of course, like the therapeutic development, is still extremely complex, and there's all kinds of interactions you have to worry about. So, no, we don't have something in the back. But this is what we want to do systematically for every variant in the human genome. We want to build these resources so that it's just a matter of computation to then be able to integrate them to infer these circuits. Here's another example. That's the strongest genetic association with Alzheimer's in this particular case. So the ApoE4 allele, when homozygous, increases your risk for Alzheimer's by a factor of 10. So this is enormous. So what we showed is by single cell profiling, if you look at all of the cell types in the brain of ApoE4 carriers versus E3 carriers, what you basically see is that cholesterol biosynthesis goes up in the carriers. So somehow they're making more cholesterol. But what's really weird is that their myelination is actually down. So cholesterol is, of course, helping myelination in oligodendrocytes that then protects the axons of the neurons. So how can myelination be down if cholesterol is up? Our hypothesis is that cholesterol biosynthesis goes up to combat the lack of myelination, but there's a problem, which is the cholesterol gets stuck. The transport doesn't work. You basically see this lipid accumulation in the endoplasmic reticulum of the oligodendrocytes that would normally be able to shuttle this cholesterol and form these beautiful myelin sheaths, and is now able to, is now stuck. So you basically have this lipid damage in addition to the lack of myelination. So we reasoned that the problem is probably transport. Again, apolipoprotein E is a transporter. So that makes complete sense in retrospect. And indeed, we said, well, wait a minute, cyclodextrin can be coupled to cholesterol and it maybe help the transport. And indeed, that restored myelination and that also restored cognition for the mice. So by, again, understanding the circuitry, being able to trace a genetic locus to a circuit and to understand where to put the X, we were able to intervene. And then again, we are now testing this at the subsequent levels of development. Here's a third example with cancer, where I'm basically going through the major killers here. So basically between Alzheimer's, obesity, and cancer, I think we got things covered. But in this particular case, we looked at responders to immunotherapy in collaboration with Geneviève Bolland and David Liu, both here at Harvard. And what we basically found is that responders versus non-responders show dramatic differences in their epigenome again across hundreds of regions. How do you fix hundreds of regions? Well, you look for the upstream regulator. And predicting the upstream regulator and using a combination treatment of immunotherapy plus targeting that regulator, we were able to basically show 100% efficacy in the treatment instead of the 50% that we currently have for metastatic melanoma. So these are 3 examples across a range of disorders to basically give you a sense of where we want to go. We basically want to systematically understand every single one of these genetic loci. And of course, some are going to be much harder than others. and be able to understand the complexity of disease. And in all of those examples, I showed you, a very simple story, but of course, it's all simple in retrospect. The stories are much more complex when you're in the midst of the puzzle. And that's where massive data comes in. That's where understanding the single cell circuitry is extremely important. So being able to see within your data every single cell and where it sits based on its transcriptome. So here the dots are positioned, the dots are cells, 20,000 dimensional vectors, and they're positioned according to their transcriptome, their global transcriptome. So basically what you're asking is what is similar to what in the 20,000 dimensional space, and then you project that down in a two-dimensional space using a neural network that converts one space into another, that warps it, such that the 30 closest neighbors of every cell are the same in the 2D space as they are in the 20,000D space. Okay, so that's what we call an embedding. An embedding is a lower dimensional projection, a manifold where very high dimensional data lives, but where you can kind of walk around in a much more natural landscape than sort of traversing these 20,000 dimensional vectors. So in this space, we can basically recognize different cell types, and we can also recognize different cell states. So basically, there's different states of excitatory neurons between 6, 2, and 4, etc. And those states are actually differential with amyloid pathology. So some excitatory neuron states are more associated with amyloid and others less. What's really interesting is some of them are also more associated with female donor cells and others with male donor cells. And in general, we found this systematic association between neurons and microglia, between these disease-associated states and these female-associated states, suggesting that, in fact, female donors had a stronger transcriptional burden than male donors. And in fact, we found that there are thousands of genes that are differentially expressed between male and female individuals before Alzheimer's, but in fact, thousands more after Alzheimer's. So our brains, of course, start out different with a different circuitry of male and female individuals, but they become even more so in the presence of disease. So we have to think about sex as a biological variable in everything we do, including treatments. And in particular, we found that the myelin that we saw earlier was in fact dramatically different between male and female individuals. Male individuals were able to upregulate myelination pathways much more effectively than female individuals, perhaps combating the loss of myelination, when in fact, and in fact, we predicted that there would be increased myelination loss, increased white matter loss in the female individuals. And post facto, we looked at the pictures that had been taken from these individuals, from these donors while they were alive. And indeed, we found that there was a dramatic difference in myelination between male and female individuals. There was a lot more white matter loss. Again, the transcriptome is guiding the phenotype. So by profiling things in a completely unbiased way at the single cell level, we're able to make predictions at the pathway level that we can then recapitulate functionally. Another striking finding that we saw was that if you compare the non-AD individuals that you see here, so there were 24 that showed very few pathological signatures and very nice cognition, compared to the mild cognitive impairment individuals and the severe AD individuals. So then we compared this group and that group. And what we found is that early in the disease, you find a lot of extremely cell type specific changes. So these are the prodromal changes, if you wish, the hallmarks that initiate Alzheimer's. But late in the disease, we found a lot of sort of shared changes between the different cell types, suggesting perhaps that this is maybe the late stages where you have all of this damage and all of these loss, and you're just fighting a battle at loss, if you wish. So maybe this is the place to start understanding the place of intervention and maybe even prevention, maybe even for some of those ApoE4 carriers to sort of start seeing a much cleaner set of pathways. And maybe this is the place that you want to sort of, I don't know, repress the fires, if you wish, at the end. Since that early paper that was published in 2019, we basically published just last year another paper in Nature that now looks across 7 different brain regions. And that allows us to now start observing the progression of Alzheimer's that has this stereotypical set of regions that get affected one after the other. So we now have many, many, different subtypes of of interneurons and excitatory neurons, many more subtypes of even glial cells. So basically, we can now see astrocytes that are dramatically different between the different brain regions. For example, the mammillary body astrocytes or the thalamus astrocytes or the angular gyrus astrocytes are very different from each other. And you can also start tracking AD progression across these millions of cells by building a pseudo time progression time course of Alzheimer's based on the prediction at every single cell for how Alzheimer's-like is that cell. And that allows you to now find what are the genes that are the most associated with early versus middle versus late stage progression. And again, start thinking about places of intervention for each of those. You can, of course, go a lot further. And instead of just studying every gene in isolation, you can now start studying combinations of genes. So we can take this giant expression matrix of 20,000 genes by 2.4 million cells and look at this lower dimensional projection into a small number of modules. So what I'm showing you here is this ability to discover which genes are co-regulated with which other genes. Because that view gives you so much more robustness, because any one gene in isolation in a single cell can be measured in only a noisy way. But if you start looking at an entire pathway, then you have much more robust measurements. So you can now use that to start understanding specific modules of genes that are dysregulated in the context of disease. And you can also start understanding subtypes of these cell types. So again, we often think about, of course, subtypes of neurons, but here I'm showing you subtypes of astrocytes. And in fact, every cellular neighborhood in this two-dimensional manifold is associated with different prevalence of different brain regions, but also of different modules that you're showing here. So if I basically place every cell here, numbered 1, 2, 3, 4, 5, 6, etc., then you can see the expression of those cells across the different genes that are associated with these modules. And you can basically start painting different neighborhoods of function, not just transcription, because you can see what are the genes that are expressed in each one of those. And what we found is that one of those was in fact dramatically altered in resilient individuals. So these are Alzheimer's donors. So Alzheimer's, so donors in this Alzheimer's cohort that show all of the phenotypes for Alzheimer's. They have massive plaques and tangles, but they were never diagnosed clinically because they have preserved their cognition. So that allows us to say, okay, what's different in those individuals? And what we're finding is that astrocytes and also oligodendrocytes are basically associated with very specific changes in genes associated with choline metabolism. And in fact, in the paper that we just published with Li Hui, who will be actually the closing lecture this year, what we showed is that restoring choline is in fact showing therapeutic effects. In the paper that we just published again in Nature with Li Hui's lab, in the context of ABCA7, another one of those lipid dysregulation genes. So to be added to your list, David, if it's not there already for your secrets to longevity. So choline appears to have a protective effect in sort of maintaining your cognition even in presence of neurodegeneration. So in the paper that we just published in Cell a couple of weeks ago, we basically now profiled not just the transcriptome across these millions of cells, but also the epigenome across the same cells. So basically, we are now looking at matched single-cell attack and single-cell RNA, in some cases from the same suspension, and in other cases from the same exact cell, where you can use the same barcode to now start matching the transcriptome and the epigenome. The first thing that we found is that if you measure the preciseness of epigenome positioning, how? Using this very simple metric of what fraction of reads from my single-cell attack data lands inside these candidate cis-regulatory elements, or CCREs. We're just simply saying, how compact is it? Are the reads diffuse across the genome, or are they super precisely positioned in peaks? And that measure of epigenome information content, if you wish, so the opposite of entropy, you could call it information, what you basically see is that it peaks in this microglial activation trajectory during the progression of AD. And then for those individuals that never reach late AD, that sort of stay in only moderate or even non-AD, it sort of stays high. But for those individuals who ultimately develop severe AD, you basically see this progression falling back down. So somehow, as you progress in microglia activation, that's when you line up your epigenome, you express the genes that matter, and that's the microglia activation part. But then you can maintain that battle, if you wish, for the non-AD individuals, but lose that battle, and then the epigenome gets diffused again through loss of information. And what's really interesting is that we also looked at the enrichment for genetic variants along the same activation trajectory through the pseudotime of microglia. And what we found is that the genetic variants associated with Alzheimer's show the strongest enrichment exactly there. So these might be the variants that allow you to sort of transition to this maintenance state instead of falling back down. So again, like we don't have answers. Our paper raises, unfortunately, more questions than it answers, but we have tantalizing hints that the preciseness of epigenome maintenance might actually be providing such an epigenomic signature of resilience again. I guess I'm going to get shot for this figure. This is basically showing you in 100,000 base pair chunks, the entire genome measured across multiple brain regions shown here at the top, the entorhinal cortex, the hippocampus, thalamus, anterior gyrus, prefrontal cortex, etc., temporal gyrus. And then it's showing you also the cell types right below, between excitatory neurons, inhibitory neurons, etc. And it's also showing you whether those cells came from control individuals in blue, early Alzheimer's in pink, or late Alzheimer's in red. And if you look here, there's a big chunk of the genome that is happy, merry-go-lucky, nothing happens. But in different cell types, as you look down here, and in different cell types, and in different brain regions, you start seeing these clusters of alteration. So for example, here, if you see the column that tells you whether those individuals are AD or non-AD, you see that there's chunks of red, even though this was clustered completely unbiased. So those chunks of red are in fact showing you specific regions that are now activated in Alzheimer's when they should be repressed. And you can see that these regions are in fact sometimes falling in vascular cells in this particular case. So again, vasculature changes are thought to precede some of the later changes in Alzheimer's. And those are in fact happening within these lamina-associated domains. So because we've colored these 100,000 chunks of the genome, you can also color them based on the cytogenetic bands. And what you can see is that these are the darker cytogenetic bands that are sitting at the periphery of the genome. So what's happening there? What's happening is that these regions are basically going from repressive to active in the vasculature. So basically, they're going from this dark blue to the pink. So that basically tells us that the lamina-associated regions are actually detaching from the lamin, and they're becoming activated. And again, you can see these progression happening in every cell type. So to the left here, I have excitatory, inhibitory, vascular astrocytes, OPCs, etc. And for each of those, you can basically see these regions of detachment that are happening over and over and over again. And you can also see the chromatin states associated with these regions, and basically see how over here you have these active regions, and here you have these lamina-associated domains that are in fact sitting in these quiescent and these heterochromatic states. So we basically are able to, across the entire genome, through this massive data integration, see the data just jump out at us. And then the way that we're interpreting is that there's these sort of non-linear changes in these transitions with this epigenome information, and that this is differentially affecting different cell types. Here I'm showing you the cell type specific information loss between cognitive resilience at the top on the y-axis and AD pathology on the x-axis. And what you can see is that the two track inversely, of course, with each other, that resilience continues to be high, and that in this information content of the epigenome appears to be helping, if you wish, both for pathology, but also for resilience despite pathology. And again, we see these changes where the differentiation becomes very precise. That's when information gain happens. And eventually, you have this fuzziness that happens in late AD, and that's where you lose that preciseness. So you can now start interpreting the epigenome and start saying, well, are all these regions that are changing in the context of disease things that we've had forever? I was just having a wonderful meeting taking back to my old days on yeast. And, are all these Alzheimer's regions something that you can study in yeast, or are they perhaps recently evolved in the context of human? So one of the things that we can now do with deep learning is that we can basically go and study not just sequence evolution, but now we can study functional evolution. And in particular, we can assemble all of these genomes across modern humans, archaic humans, great apes, and then start predicting using convolutional neural networks and these sort of prediction functions, the actual activity, the epigenome activity for every one of those, and identify regions that are differentially accessible between modern humans, archaic humans, hominins, and primates. And what we're finding is that there are dramatic changes that we are predicting computationally and that we can validate experimentally. So we basically actually used measurements of the epigenome change in great apes versus hominins, and we found that indeed the deep learning predictions are matching these changes. And we found some dramatic alterations. So basically across the entire genome, we found 40,000 hominin-specific changes and 700 Neanderthal-specific changes, including this one example here, where we basically have these KCNS1 neuronal acceptability potassium channel in the CNS that is also, at a talk that I just heard from Svante Pabo at the Museum of Science, is also showing recent protein coding alterations in modern humans versus Neanderthals. So again, you can kind of see these changes that we can now use to sort of map these disease associations directly onto the genome. And we're finding these striking examples of gains and loss, thousands of events, and we can now, using all of the genetic data, ask where are they implicated? And what we're finding is that the vast majority are in fact associated with neurogenesis. So indeed, we didn't just fall off a tree, we dramatically altered our neuronal projection and so on and so forth, but also regulation of locomotion, organ development, cell communication, cell adhesion, and so on and so forth. So that allows us to now bring this evolutionary eye. We're also using spatial methods to understand not just brain, but also heart. In this particular case, again, one of the greatest killers of our planet is coronary artery disease. And what we're basically finding is that these lumen-specific cells that are immune cells that are capturing lipids once more inside our blood are basically showing very specific spatially constrained changes within the lumen that again allows us to start inferring what are the right cell types and what are the right genes that we should be targeting. We're also looking at how obesity and diet and exercise are interplaying with each other in the context of, again, another collaboration here with Harvard, with Laurie Goodyear's lab, with Jackie Yang, that just started her own independent faculty position, Maria and Leandro, that is basically showing us how exercise is in fact affecting the brain of these mice in this particular case, where we basically are finding that obesity training and rescue are having very specific changes within the brain, very often affecting the interface between neurons and other cells, and in particular immune cells and oligodendrocytes, which is, again, fascinating. We're also able to study heterogeneity in the context of Alzheimer's across now 400 individuals that we published last year in Cell in 4 back-to-back papers, again in collaboration with Li Hui, where we basically found very specific cell types that are associated with resilience, and where we can now start understanding the heterogeneity of those individuals. And in particular, what we're finding is that Alzheimer's doesn't affect random genes in random people. there's a small set of hallmarks that are repeatedly altered. And we can now use our single cell data to sort of start mapping these recurrent changes in Alzheimer's and start identifying specific hallmarks where we can predict exactly which pathways we should be targeting for each individual, because only a different subset is regulated each time. So in a paper that was published in Science last year, we basically looked at schizophrenia. And in particular, we found that if you try to predict, as I just mentioned, schizophrenia from the transcriptome, you can do a very good job. You can basically say, great, I can predict schizophrenic donors versus non-risk control donors. And at the two ends of the distribution, the prediction is very, very good, except for asterisk, this person here. What is this person doing there? This is the fifth most schizophrenia-like transcriptome, and it's found in a control individual. And it's not just one cell, this is across all of the cell types of that person. So what could be going on? Well, that person, it turns out, has an asterisk because it's not just a control. This is a donor whose son was diagnosed with schizophrenia. And given the 1 in 1,000 prevalence of schizophrenia, you would expect that maybe there's some transcriptional changes here that we're picking out that are indicative of perhaps an increased risk that then manifested in the subsequent generation. Conversely, we have schizophrenia cases here that are showing some of the most control-like transcriptomes, and those are in fact showing an abundance of this SZTR cellular state, suggesting perhaps a different path to schizophrenia that only affects a subset of those cells. So that got us to thinking maybe we shouldn't be thinking of donors monolithically. We shouldn't be saying that every Alzheimer's person is 100% Alzheimer's and every control is 100% control, which is what we're actually doing now. So every single cell differential expression analysis method is basically assuming that all of the cells from a control are control cells, which is a ridiculous assumption, and that all of the cells from an Alzheimer's donor are 100% Alzheimer's, which again is a ridiculous assumption. So what we basically developed is a new method that we call cell-projected phenotypes. What do? Because we have 400 donors, we can now start looking at every neighborhood in this latent embedding landscape of transcription. So every transcriptional neighborhood, and then ask, are the donors of those cells in that neighborhood mostly Alzheimer's in red, or mostly controls in blue? So now we can find Alzheimer's neighborhoods and control neighborhoods. And we can now start asking, what are the genes that are different in this neighborhood versus that neighborhood? And between this and that neighborhood, what are the changes that are happening? So we can now recolor every cell, not based on the 100% red or 100% blue color of their donor, but instead based on the gradient of how red or blue are the neighbors of that cell. And now instead of saying this person is Alzheimer's, we can forget the original phenotypes of that person and say this person's microglia are sitting in Alzheimer's states, but that same person's oligodendrocytes are sitting in control states, and not just in microglia, not just in Alzheimer's states. You can say in Alzheimer's lipid dysregulation states or Alzheimer's myelination dysregulation state, and so on and so forth. Is everybody with me here? So that's so exciting because I can now take every single phenotype associated with Alzheimer's. I can take tangle density or cognitive impairment or AD and start painting every cell from every person, every cell type from every person, based on where the cells of that person are according to the painting of that phenotype onto all of the cells from that person. And then what I can see is that there's a gradient, not just black and white, not just red and blue, but there's a gradient. And indeed, some individuals that are very blue appear among the red. So now we can say, well, maybe that person's microglia are the problem rather than their oligodendrocytes. So that allows us to now start discovering trajectories through this cellular space and discovering differentially expressed genes at unprecedented resolution. When before we had 3 or 4 genes that were becoming significant, now we have hundreds of genes that we can find as differentially expressed, even though the overall correlation between the measured and the inferred doesn't change. The subtle changes are in fact leading to dramatically higher discovery power. And we can now start also asking for intervention. Where do I go to intervene to bring the cells from this person from one sick state into a healthy state? And that's what takes us to now taking these hallmarks that we're identifying at the cellular level and then using the genetics of each individual to start projecting the risk of that person on each of the hallmarks. So instead of just saying, oh, sorry, you have a high risk for Alzheimer's, we can say you have a higher risk for myelination problems or neuroinflammation problems or neuroenergetic problems and so on and so forth. So we can now calculate the burden of every individual across both regulatory variants and protein coding variants using all of these deep learning methods and this extraordinary technology that we have, both from the experimental results and the AI results, and start converging all of this burden at the level of every gene and at the level of every pathway and at the level of every cell type. So we can now start predicting from the genetics alone the burden of each person. That allows us to now start moving towards, of course, precision and personalized and predictive medicine, which I think we're all trying to achieve. And just in the last few words, I just want to say that we can now use these techniques to start training AI models that can span all of these different modalities, that can basically understand human language through large language models. And we can look at the corresponding embeddings, understand the biology with specialized biological language models, understand the single cells through foundational single cell models, understand the geometry of both the protein structure and the chemical structure, and understand how to map structure to function through fine mapping of biological annotations onto structural domains and motifs. And we can now build these reasoning, interactive, user-guided, and AI-powered drug design systems that are basically able to reason with us, talk with us, speak both English and protein structure. So we're using that to now start designing new therapeutics that are able to bind the specific pockets that we are predicting from this exercise. And we're building this agentic drug discovery pipeline that is allowing us to now reason through agents that are able to carry out the work of many of our analysts and say, let's go and, so the prompt is find a combination of approved post-phase 1 clinical trial drugs that can treat Alzheimer's. So 15 minutes later, we have the answer. And it basically says, okay, I'll begin by searching for cell type specific mechanisms. I'm searching my corpus. Now this literature indicates those. The targets must be differentially expressed. Great, so I'm going to use a differential expression tool. I will now proceed with their validation. So I'm looking at the modules. I'm finding that they're not pathway redundant because, we're looking for a combination treatment. So I will now check the tissue expression to make sure that there's no off-topic, off-target effects. So now I need to identify the biomarkers. I'm reading the literature. I found the biomarkers. Great. Now we'll now search for drugs that are suitable for these particular targets. So it's selecting the drugs by looking at the chemical landscape that we have constructed through these embedding representation learning. It's now finding the drugs and it's docking them. And then at the end of these 15 minutes, we basically have candidates that have unbelievable specificity. So if you look at the molecular specificity of those candidates, specifically in the pockets that we care about, they are incredible. So here you can see the agents at work, basically carrying out their function. So after seeing this, one of my computational grad students says, Manolis, I want to learn how to do experiments. And I'm like, wait till you see our robots. So what I convinced him to do instead is to train the agents rather than sort of fear them. So we are now working, here's the robots, we are now working with Brad Penteluta's lab to basically now synthesize both peptides and small molecules. And our agents are able to now not only synthesize drugs that we should be making, but also how to make them efficiently, which we can now use within these collaborations, now look at all of these different therapeutic modalities. And of course, in collaboration with Leeway, with Bob Langer and other extraordinary collaborators, we're now able to test these drugs directly into organoids. And we are now collaborating with a clinic that is looking at hundreds of patients that have predispositions, risks, and sometimes early manifestations of Alzheimer's. So we can now grow organoids that are matched between the patients and the brains. So basically, we now have postmortem brains and brain organoids that are matched. We are already carrying out that analysis, and we're now looking at imaging biomarkers, blood biomarkers, et cetera, to be able to do this. So unfortunately, I got through only the first part of my talk to talk about big data multiomics, understanding the circuitry, then using AI for drug design, and then screening the organoids, and then optimizing chemicals. Should I show you a quick preview of the next one? You guys are maybe 2-3 minutes. Very quick preview. Okay, very quick. So basically, the last point that I want to make is about cognitive cartography, being able to map the landscape of function and being able to navigate it directly. So we have built this unbelievable interactive interface that we call Mantis, because it's the seer that sees beyond, that is able to now, for example, here, look at 20,000 human genes in their embedding that is learned through protein structure and function. And you can now search. You can see the results directly on the map. You can browse the ontologies and see different parts light up. You can talk to the agent and say, hey, find all of the genes associated with Alzheimer's through the literature. You can actually plot, and you can also ask the agent to write analysis that will be run directly onto this landscape. So we're using this to understand how to consume information completely different ways. So this is, for example, scientific podcast where you can, I don't know, search for AI or meaning of life or, Manolis Kellis, or you can navigate our own research ideas. So every idea that we've ever had in our lab is now recorded and is traceable and searchable. So I can click on every person from our lab and then see that person light up in every idea that they've had in this cognitive space. I can click on every meeting and light up similarly. And I can say, okay, what are the key contributions of, for example, Pascal and so forth. And you can kind of click and directly browse and navigate the space. You can see quantitative information directly. This is an example for startups where you can basically see correlations of different sectors. It's able to pull information from the internet directly. Here, I'm showing you 60,000 papers published by MIT in one landscape, and then every grant from the same authors in another landscape, and also the physical map of campus while we're at it on a third landscape, and you can kind of see how information flows across them. You can see how information changes over time. You can see the relationship directly there. You can plot quantitative information there. You can see, again, how my students are progressing through knowledge space. You can study startups. You can also use agents by basically defining these workflows in a fully interactive way, combining embeddings, knowledge graphs, agents, and these tools. You can also program. You can basically tell it, hey, why don't you make me a new interface that does that? And it understands its own code. So it's able to sort of create new interfaces that are interactive and using the results that you're able to compute. So it writes the code and creates these new modules automatically. And we're using all of these to understand these multimodal representations, where you can basically go across images, for example, medical images or DNA sequences, and all of these deep learning, like alpha genome representations of the genome and its modules and its building blocks, understand chemical and protein structure, understand patient records, understand knowledge graphs and the embeddings that we learn by graph neural networks within these spaces, understand, of course, scientific papers, cells, and of course, this multimodal information. And you can use Mantis to now navigate the protein structures of every gene in the genome and the chemical structures of hundreds of thousands of molecules, and design new molecules by sampling from the latent space. You can just say, hey, there's a lot of molecules associated with Alzheimer's therapeutics here, sample more from that. And we're able to sort of map patients there. We're able to now start understanding protein structure, understanding chemical structure, mapping the two on top of each other. So this is chemical floors and protein floors mapped together. Of course, cellular spaces. And there's this programmatic automation, this agentic automation, and the road ahead is using AI not just to answer specific questions, but to look at it visually as a Google Maps, to basically navigate, not just create these embedding landscapes, but actually systematically navigate them. And I don't want to just give a magic carpet to everyone. I want to give you a dashboard where you can see your data in completely new ways. That's where I'll stop and take any questions. Thank you.
The common variants that are linked to various diseases mean that they've been inherited through quite a lot of generations, which means they've been probably selected for that are not related to the disease. So do you get any insights onto what these functions are doing, not what these functions of these are in the reason why they were selected, because obviously any common variant must have been selected for something. So
Yeah.
sort of like the way in sickle cell disease.
Yeah. So I have a slightly different view. I think that I think that it is hard to keep a variant at zero. That basically selection only kicks in after a little while, and that most of selection acting in the human genome is purifying selection. I think that for a variant to stay at like 50% frequency, that's where you see selection. But at like 5% frequency, this is simply lack of purifying selection. And in this particular case, the reason why common variants have weak effects is exactly what you're saying. It's that there's selection acting, of course, on the entire human genome, but it's only when you have strong effects that it is maintained at very low frequency. That's why strong effect protein coding variants are only showing, 0.1% frequency. Whereas the high frequency variants that we're finding, 93% of them are sitting in non-coding regions. So basically, the ones that we have are the ones that escaped selection. Why are they exciting? They're exciting because these are things that thou shalt not touch on the strong effect, but on the weak effect, they give us entry points for disease. Notice this particular variant only increases your weight only by 1 standard deviation. But when we make that edit in an otherwise isogenic, sample, we see a 7-fold change. And when we perturb the gene, we see, a dramatic alteration even at the organismal level. So I think that there are strong effects at the molecular level, weaker effects at the cellular level, even weaker effects at the tissue level, and tiny effects at the organismal level. And that's why these common variants have escaped selection. So however, you're absolutely onto something, because this particular variant is at 42% frequency in Europe, 2% frequency in Africa. So when we were in Africa, maybe we had plenty of food. And when we started crossing, I don't know, the deserts to get out, or ice ages, or you name it, maybe there was selection for being able to store calories that may have pushed it to 42% frequency. It could also be drift, but we can make a story for selection. And in fact, prior to 1942, if you look at the banked samples and phenotypes from the Framingham Heart Study, the FTO allele has no effect on obesity. It only has an effect on obesity after World War II, when we started having office jobs, McDonald's, fast food, cars, etc. So basically, I think that environment and selection, of course, go hand in hand. And many of these variants that are high frequency may have been selected, yes, indeed, in a different environment, but I think the vast majority will be drift.
One more question. There is one.
I promise me, I'll make my answer in less than 30 minutes.
Yeah, real quick.
Yeah, in the subjection phenotype part, you describe how then certain cells are affected in specific pathways, and you can like map them onto it. Is it also, can you use it for discovery of saying that you haven't discovered pathways that are affected in one disease state, or like in different convenient way?
Completely. So yes, we are now basically studying those. We're looking at the gradient between them, and then we're finding completely new pathways that we did not expect. So basically, these make complete sense biologically, but they're invisible when you look at this traditional case control 100-100. And I think that that's how we're going to get to the hallmarks of disease, that every one of those centers is going to be a different hallmark, and that the trajectories between them are going to tell us where and how to intervene through these trajectories here.
Thanks, Manolis.
Thank you so much.
It was super impressive.
Thank you. It's always a lot of fun.